this is another important section in general physiology. That's transport processes. Transport processes means how the substances enter the cell or leave the cell by crossing the cell membrane. Now basically I have divided it into two parts: transport across a cell membrane and transport through a cell membrane. What is the difference? If a substance is entering the cell or leaving the cell by invading or going through the membrane, it would be called as transport through the cell membrane. Whereas at some other instances, substances do cross the membrane, they go to the other side, but without actually going through the membrane, without actually invading the membrane. So a substance which is crossing the membrane but without actually going through the membrane or invading the membrane. Yes, that's also possible, and such transports would be called as transport across the membrane but not through the membrane, right? And therefore the examples of transport across the membrane would be endocytosis and exocytosis. These are also called as vesicular transports because the transported substance is inside a vesicle while being transported. So vesicular transports, for which one more name has been used. It has also been sometimes called as cytopempsis. It is said to be a part of transcytosis. Transcytosis. That is, substance is not only crossing one membrane, but it also crosses the cell on the other side. So, transcytosis, across the cell, through the cytoplasm, across the cell, that's transcytosis. So, these are some of the nomenclatures which are used. Transport through the cell membrane, we will be mainly dealing with osmosis, diffusion, and active transport. So that is basically a division in these transports. So, but remember, when we say transport across a membrane, that means not through the membrane, but still crossing the membrane. And then the other type is transport through the cell membrane. So let's now look at them one by one. What all questions have been asked just before we start this? What all questions have been asked on this particular segment? The questions which are problem solving type questions. For instance, a red cell with an initial volume of 100 cubic microns and intracellular tonicity of 300 milliosmoles per liter. This red cell was suspended in a solution having the tonicity or osmolarity, let's say, the osmolarity of 100 milliosmoles per liter. So this is a red cell with initial volume and its intracellular osmolarity. This red cell was suspended in a solution having the osmolarity of 100 milliosmoles per liter. How much will be the final cell volume? And you have to solve this particular problem. Look, because of the different osmolarities, there's going to be movement of water either into the cell or out of the cell. We will see that later. And finally, there would be some cell volume. So the question is, how much will be the final cell volume? There was one more question which was a clinical question. A patient of non-ketotic hyperosmolar coma, diabetes has two types of complications. One is a diabetic ketoacidosis when ketone bodies are formed, and non-ketotic hyperosmolar coma such a patient was brought to the hospital, and the findings, the lab investigations, they were something like this. Serum sodium was 145 milliequivalents per liter. Then glucose was 180 milligram percent, and BUN, blood urea nitrogen, let's say 28. So what is the plasma osmolality of this patient? Look, we have already said this is non-ketotic hyperosmolar coma. So plasma is going to be hyperosmolar, but how much? What is going to be the osmolarity? How will you calculate it? So such questions have been asked in the recent past in various entrance exams. With that in mind, let's start the discussion. Transport across the cell membrane, but not through the cell membrane, as we have already mentioned. Well, the first one, endocytosis. Many substances enter the cell, they are internalized by this process called as endocytosis. But the question is, what type of substances? They choose this method. Large molecules. The point is, the substances, they cannot diffuse through the cell membrane. They cannot use diffusion as the transport modality. Such substances will use endocytosis to enter the cell. So large molecules, macromolecules, particulate matter, particles, foreign substances, bacteria, such substances do enter the cell by endocytosis. Two types of endocytosis based on what type of substance is entering the cell. Based on that, it's called either phagocytosis, when solids are entering the cell, and pinocytosis, when liquids are entering the cell. So that's one way of classifying the endocytosis. And therefore, since solids and liquids, that's how it's been differentiated. Phagocytosis is also sometimes called as cell eating, and pinocytosis is called as cell drinking. What are the examples? For phagocytosis, if you have already read the pathology few initial chapters, you will know the bacteria entering the neutrophils. This is a process of phagocytosis. Pinocytosis, proteins in their dissolved forms, soluble proteins in their dissolved forms, they enter the cell in a liquid form. That is, that entire solution, I mean to say, it enters the cell by pinocytosis. Apart from this, there is one more way of classifying the endocytosis based on the mechanism. So what are the types? First one, constitutive endocytosis. Constitutive endocytosis means it occurs continuously. It does not need a particular signal to arrive. It does not need any such specific mechanism. Well, let's just draw here how the endocytosis occurs. That a substance to be internalized, it comes near the cell and then it will cross the membrane. In constitutive endocytosis, there will be a continuous process. By virtue of constitution of the cell, it has that ability to internalize the substance. So example, vitamin uptake by the cell, vitamin entry into the cells. Many of the vitamins, they enter the cell by constitutive endocytosis. You know, vitamins are the cofactors required for various enzymatic reactions in the cell. So almost all cells are going to need those vitamins, and therefore it's a constitutive endocytosis. Second, receptor mediated endocytosis. That is, when a particular ligand arrives at the cell, first thing that happens is there are specific receptors on the cell membrane which are identified. Those receptors will be identified. So this ligand now binds with its specific receptors. Next, this pit in the membrane, look, this is called as clathrin coated pit. So there is a fibrillar protein in the membrane, long fibrils. So fibrillar protein in the membrane called as clathrin. And this clathrin mediates this process of endocytosis. The receptor mediated endocytosis is also sometimes called as clathrin mediated endocytosis. Now, when this pit has the receptors and the ligand combines with those receptors, then the pit invaginates. So this has got bound to the receptor. Now the pit invaginates, that is, it becomes deep, and the substance having entered that invagination now here. So substance has entered this invagination. Finally, a collar will be formed around the neck region. Now this substance has entered the invagination. Now this collar is formed around the neck portion of this invagination. And then this part of the membrane will be pinched off. This part of the membrane will be pinched away from the membrane, so that a vesicle is formed inside the cell. The membrane will become normalized once again, and the substance internalized. It's inside the cell now, but within a vesicle, right? So we have already mentioned that this is a vesicular transport. Here is the internalized substance. Well, just remember, when the vesicle was being formed, there was a major role for clathrin. There is a clathrin triskelion. Triskelion means with 3 legs. That clathrin cage surrounds the vesicle. Clathrin triskelion surround the vesicle. And then eventually the vesicle has been formed. Now the next would be that this vesicle has to fuse with some target organelle inside the cell. Maybe lysosomes. And before that happens, before that fusion happens, this membrane of the vesicle fuses with the target organelle membrane. This clathrin cage has to dissolve. Yes, only then the fusion will happen. And the dissolution of clathrin cage needs ATP. So remember, for this process, ATPs are needed, and for that matter, it can also be considered an active transport type of process, just because ATPs are being consumed here. So remember, that's how clathrin mediated endocytosis happens. Receptor mediated or clathrin mediated endocytosis. What are the examples? LDL entry into the cells and LDL receptor also. Both these are internalized by this mechanism. So let's just write it. LDL entry. where LDL entry into the steroidogenic cells. You know, steroid hormones are synthesized from cholesterol. So let's say this is a steroid synthesizing cell in the adrenal cortex. Now it has to capture the circulating LDLs, and then those LDLs will enter the cell. LDL cholesterol will provide for the synthesis of the steroid hormone. So this cell expresses the LDL receptor, and then circulating LDLs will bind with those receptors and internalized, and then steroid hormones will be formed. That's a very famous example of clathrin mediated endocytosis. Well, there is one more type of endocytosis. Sometimes in some cells, instead of clathrin, there is another protein called as caveolin. So the process will be called as caveolin mediated endocytosis. Instead of the fibrillar protein clathrin, there would be caveolin. In fact, many cells, their membranes have very small depressions, and they are called as caveolae. And those depressions, the caveolae, are lined with this protein called as caveolin, and it will internalize some of the substances. The commonly given example is folate entry into the cells is by this particular mechanism, and this mechanism has also been called as podocytosis or podocytosis. Some authors call it podocytosis, some call it podocytosis, but that's how folic acid enters the cells. Right, that was how the substances are internalized. These were the types of endocytosis. The other transport across cell membrane is exocytosis. Exocytosis is the one in which the substances will be thrown out to the exterior of the cell. Now, there are two types of exocytosis. One, constitutive exocytosis. Well, first of all, just I may add here, exocytosis is also called as cell vomiting. Cell vomiting. You know, we have seen some time back cell eating, cell drinking, now cell vomiting. And of course, you must have read about the apoptosis and necrosis, which is called as cell suicide and cell murder. Right. So exocytosis, again of two types. Constitutive exocytosis is the one which occurs continuously, as a continuous process by virtue of constitution of the cell. What are the examples of constitutive exocytosis? Immunoglobulin secretion by the plasma cell. You know, B lymphocytes, they are known to secrete the antibodies or immunoglobulins. But how does that happen? After the antigenic exposure, the immunoglobulins or antibodies are formed by the B cell. And when those antibodies are to be secreted, the B lymphocytes form an intermediary cell called the plasma cell, and then the plasma cell will continuously secrete the antibodies. Okay. This will be a continuous secretion after that. Or any other example, yeah, mucus secretion by the mucus secreting cells. I mean, let's say goblet cell in the duodenum. This is also a continuous process of exocytosis. Mucus lining will happen because of that continuous secretion. The other type of exocytosis is called as regulated exocytosis. Most of the examples of exocytosis, they fall in this category, regulated exocytosis. Regulated means it will not happen continuously. There will be some kind of a signal that will arrive, and under the influence of that signal, there would be exocytosis. Neurotransmitter secretion at the synapse, acetylcholine secretion at the neuromuscular junction. These are all the examples of regulated exocytosis. This is a typical synapse, and there's a neurotransmitter located inside a vesicle. We have already said that this is a vesicular transport. Impulse arrives at the terminal. Because of the impulse, the voltage gated calcium channels will open. Calcium will enter, and then the vesicle will travel towards the membrane, presynaptic membrane. It will fuse with the membrane, and finally, by exocytosis, the neurotransmitter will be thrown out. It will be thrown out into the synaptic cleft. So that's regulated exocytosis. Right. I may just add a point here. Calcium came in, so calcium was needed for this process. Why calcium was needed? Because the vesicular membrane and the target membrane both are negatively charged. Then they will repel. There will be repulsion. So there has to be some bridge formation between the two membranes. And that bridge formation between the two membranes means the vesicular membrane and the presynaptic membrane. The bridge is formed with the help of that calcium. So calcium influx necessary there. That's the example of regulated exocytosis, or let's say hormone secretions. Most of the hormones are secreted by regulated exocytosis. Insulin secretion by the pancreatic beta cell. That's a regulated exocytosis. Right. So these were the types of the exocytosis. Finally, let me just make one point. That is, all the processes of exocytosis, they need an increased intracellular calcium. Right? We saw that just now, that calcium comes in and only then the exocytosis happens. So increased intracellular calcium precedes the exocytosis. This happens, and then because of it, the exocytosis happens. So this is true for all the processes except at two instances where decrease in intracellular calcium will lead to exocytosis. Okay, let me just write it here. Except in the two instances where decrease in the intracellular calcium results in exocytosis, or it is followed by exocytosis. If not results, first there is a decrease in intracellular calcium and it is followed by exocytosis. What are those instances? Okay, parathormone secretion by the parathyroid gland. This happens with a decreased intracellular calcium. When is parathormone required? When there is a fall in the serum calcium. Now this fall in the serum calcium, intracellularly, how is it signaled? Intracellularly, the signal is in the form of a decrease in intracellular calcium. And then the cell, the parathormone secreting cell, will then release the parathyroid hormone, and it will then elevate the calcium levels. Another example is renin secretion by the JG cell. Renin secretion by the JG cell also happens by a decrease in intracellular calcium. What I mean is, first there is a decrease in intracellular calcium, and that leads to release of renin by the JG cell. When is renin needed? Or when is renin-angiotensin system to be activated is with the decrease in the blood volume and decrease in the blood pressure. This decrease in the blood volume or decrease in the blood pressure, how is it signaled inside the JG cell, juxtaglomerular cell in the kidney? It's in the form of decreased intracellular calcium within the JG cell, and that will be the signal to release renin. And then the further things, angiotensin and aldosterone, which we will discuss in the CVS chapter. So after having discussed the transport proteins in the cell membrane, now is the time to discuss transport processes through the cell membrane. We've already made a distinction between two types of transports: transport across a membrane and transport through a membrane. When we say transport through a cell membrane, it means the substance is invading the membrane, going through the membrane to go to the other side of the membrane. Whereas when we say transport across a membrane, means the substances cross the membrane. Look, basically in all the cases, the substances are crossing the membrane to either go into the cell or out of the cell. But when we say transport through the cell membrane, means it goes through the cell membrane, invades the membrane. And when we say only transport across the cell membrane, for example, endocytosis, exocytosis, in those cases, the substances, they do cross the membrane, but without actually going through the cell membrane. So, having made this distinction, now is the time to look at these processes, transport through a cell membrane. Let's begin with the first one, the osmosis. Let's see some of the basic points of osmosis. Osmosis is movement of water from low solute concentration to high solute concentration. At a very basic level, that's the osmosis. So, if we have two solutions and they are separated by a semi-permeable membrane, then the water, which is the solvent, will go from low solute concentration to high solute concentration through the membrane. So, in a way, we can say wherever there is high solute, and to understand the principles later, I would say wherever there is high sodium, water moves towards it, and that's called as osmosis. So, wherever high sodium level, it will pull the water by osmosis. Right. Now, this is sodium pulling water towards it by osmosis through the semi-permeable membrane. There is also one more type of transport, and that is called as bulk flow, or also called as solvent drag. This is opposite to that of osmosis. What is solvent drag? Water moves in bulk and it drags sodium along with it. It drags sodium along with itself. So, solvent, water, and therefore it's called as solvent drag. Water is dragging the sodium. As water moves in bulk, it is dragging the sodium along with it. Right. Make the distinction. Osmosis, high sodium is pulling water towards it. Bulk flow, water moves in bulk and it pulls sodium along with it. Where does this happen in the body? Okay, descending limb of loop of Henle. Let's see some of the examples of bulk flow or solvent drag. In the descending limb of loop of Henle, it so happens that water moves out of the tubule into the interstitium and it drags some sodium along with it. CSF reabsorption, another example of solvent drag. When CSF is formed and then it comes into the subarachnoid space and then it's going to be absorbed on the arachnoid villi, water is absorbed, or let's say reabsorbed, and some sodium also moves along with the water. Capillary filtration, another example of solvent drag. You know, in and around the capillaries, there are Starling's forces. We are going to discuss those Starling's forces later on in the CVS. But these Starling's forces, like capillary hydrostatic pressure or plasma colloid oncotic pressure, they are going to cause movement of water either out of the capillary or back into the capillary. So water will move in bulk and it will drag some sodium along with itself. So that's solvent drag. However, we are concerned with the osmosis, and we've already defined it. Wherever there is high sodium, it will pull water by osmosis. Second point, what's osmolarity? Very often we are asked, or we have to know the difference between osmolarity and osmolality and tonicity. These terms are often used interchangeably, but there is a slight difference between the terms. So, osmolarity means solute per liter of the solution. I mean, number of solute particles per liter of the solution. Whereas osmolality is the mass of solute per kg of the solvent, and in the body water is the solvent, so I have written it as water per kg of water. So that's a difference between osmolarity and osmolality. The perspective is different in these two terms. I mean, I'm talking conceptually, so look at the perspective. When you say osmolarity, you're talking about the entire liter of solution and in that the mass of solute. Whereas in the osmolality, it's the solute relative to solvent, right? So osmolality is a ratio of solute per solvent. That's the difference. So what difference would it make whether we use osmolarity or osmolality? Let me give you an example. Change of temperature will affect the osmolarity, but it will not influence the osmolality, right? Why? Because the change of temperature will influence the entire volume of the solution, liter of the solution or volume of the solution. So it will influence osmolarity. But osmolality, which is the ratio of solute to solvent, that will not change with the change of temperature. So remember, that's how you can differentiate between the two terms. Let's talk about serum osmolality. Serum osmolality, and I think at the start of the session, I had asked one MCQ. A patient of non-ketotic hyperosmolar coma. I had mentioned this, that the diabetes is likely to produce two types of complications. The diabetic ketoacidosis or non-ketotic hyperosmolar coma, when glucose becomes 700, 800 milligram percent, very, very high. And that increases the serum osmolality. So, the question was, following were the findings in that case. Serum sodium was 145 milliequivalents per liter. Glucose is 720 milligram percent, and BUN is 28. So this has been given, this data has been given. Serum sodium is given, glucose levels are given, and BUN, blood urea nitrogen, is given. And the question is, what is the serum osmolality? Calculate the serum osmolality. Let me change it further and let me ask you, calculate the serum tonicity from this. Instead of osmolality, I am calling serum tonicity. So how do you calculate that? What's the difference? Let's see. Serum osmolality is calculated by this formula. And of course, since we are talking of osmolality, it would be milliosmoles per kg of water. It's equal to 2.1 into sodium plus glucose, whatever glucose level is given, divided by 18 plus BUN, urea or blood urea nitrogen, upon 2.8. That is the formula for calculation of serum osmolality. So sodium, glucose and urea are taken for the calculation of serum osmolality. Now you know, and you can calculate and answer that question. Now coming to the tonicity. What is the difference between osmolality and tonicity? While calculating osmolality or osmolarity, we will take all the osmoles of all the solutes will be considered. While calculating tonicity, we will only take non-penetrating solutes. From all those solutes which we considered for calculation of osmolality, from all those solutes, we will only take non-penetrating, non-permeant solutes. Means those solutes to which the membrane is not highly permeable. So only those will be taken for the calculation of tonicity. So osmolality, we took sodium, glucose and BUN. For tonicity calculation, out of these three, we will only take sodium and glucose. Membrane is not very highly permeable to these two. We don't consider urea for the calculation of tonicity because the membranes are highly permeable to urea. Okay. Why? I mean, what is the basis for that? Urea crosses the membranes easily, which means what? Urea will reach equilibrium very, very rapidly between the two compartments, between the two solutions. Urea will reach equilibrium very rapidly. So, it will not cause movement of water. Remember, water movement by osmosis will be caused if the solute is higher on one side of the membrane compared to the other. Okay. There should be unequal concentration of the solute on either side of the membrane. Then it will cause movement of water from the low solute concentration to the high solute concentration. Urea, that's generally not possible because urea permeates the membrane so rapidly that it reaches equilibrium very rapidly. So it will not cause movement of water. it will not remain unequal on either side of the membrane for too long. It will quickly reach equilibrium. So urea is therefore called as ineffective osmole. Ineffective osmole, and therefore it's not considered for the calculation of tonicity. We will take it for osmolality, but if they are asking calculate the serum tonicity, we will not take urea, we will take only sodium and glucose. So remember that it's considered as an ineffective osmole. So the underlying fact that I wanted to emphasize is that if the solute remains unequal on either side of the membrane, then, I mean high on one side, low on the other side, then it will cause movement of water from low to high concentration of the solute, right? So that's the difference. So tonicity, in a way, we can say for the body, we are talking about the cells, okay? Water inside the cells and water outside the cells. So in the context of cells, we may say that tonicity is considered to be a potential to cause changes in the cell size. Why? Because imagine that a solute remains unequal inside the cell and outside the cell. Then it will cause movement of water from low to high concentration of the solute. So either the water goes out of the cell or water goes into the cell, depending on where the solute is high concentration. And that means the cell size will change, isn't it? Water will move out or in. So tonicity is a capability or potential of the solution to cause changes in the cell size. I mean, say an RBC, and inside the RBC, let's say this is the osmolarity, and outside the RBC, this is the osmolarity, or let's say tonicity of the solution. RBC is suspended in this solution with the tonicity of, let's say, 200 milliosmoles per liter. Now this is a hypotonic solution, right? Less solutes, more water. Hypotonic solution. So RBC has been suspended into the hypotonic solution. What will be the result? Water will move from low solute concentration to high solute concentration. It means water will enter the RBCs. It will move into the RBCs, and that means this RBC volume will increase. RBC size will increase. So that's the importance of tonicity, and tonicity of the plasma, tonicity of the ECF. That's how it is important. It has the ability to cause changes in the cell size. Finally, I think I had asked one more MCQ at the start of this section. An RBC with an initial volume of 100 cubic microns, that was the initial volume of the RBC, and the intracellular tonicity of 300 milliosmoles per liter, or intracellular osmolarity of 300 milliosmoles per liter. I am using the terms interchangeably here, osmolarity and tonicity. So the point is, this is the intracellular tonicity. This RBC was placed in a solution having the tonicity of 100 milliosmoles per liter. What will be the final cell volume? What will be the final volume of the RBC? So far, you must have guessed that this is a hypotonic solution. 100 milliosmoles per liter means hypotonic solution. And when RBC is placed in this solution, water will enter the RBC and RBC size will increase. But what will be the final cell volume? How to calculate that? Simple. Look, you have been given two osmolarities and one volume, and you have been asked to calculate the final volume. How do you calculate this? Simple formula. Pi I Vi is equal to pi F Vf. Alright. Initial osmolarity or intracellular osmolarity into initial volume is equal to final osmolarity into final cell volume. So three things have been given and you are asked to calculate the final cell volume. Now it shouldn't be very tough. 300 into 100 is equal to 100 into, simple, 300 cubic microns. That will be the final cell volume because water has entered the RBC. That was about the osmosis, osmolarity, osmolality and the related information. Let's move on and discuss the next transport process, diffusion. Okay, first thing, diffusion is a passive transport, right? And when we say passive transport, no ATP is needed. So that's very clear. When we say active transport, it needs ATP. Passive transport doesn't need ATP. Second, net diffusion is a downhill transport. Net diffusion is a downhill transport. Downhill means going down the hill. So it's net diffusion is from high to low concentration of that particular substance, right? Remember, I am using the term net diffusion, net transport. So the point that I am trying to make is that diffusion can occur from high to low concentration or low to high concentration, but net will always occur from high to low concentration. Don't forget that. It's a net diffusion which we are talking about, right? There are two ways in which the diffusion occurs. We are talking about the cell membrane, and cell membrane is made up of lipid bilayer and the protein channels in the membrane. So two ways. Through the lipid bilayer, lipid soluble substances, they will diffuse through the lipid bilayer, and through the transport proteins, let's say through channels in the membrane. Water soluble substances cannot diffuse through the lipid bilayer. They have those specific channels for them, right? Next point, there are two types of diffusion. Well, now basically at a very basic level, there are two types of diffusion. There are a few more which we will discuss in the end of this section. Simple diffusion and facilitated diffusion. In fact, to avoid confusion, let me just make a small correction. Through the lipid bilayer and through protein channels that we said just now, we were talking about the simple diffusion. Simple diffusion of the substances can either occur through the lipid bilayer or through the protein channels. Lipid soluble substances, lipid insoluble or water soluble substances. So these were the mechanisms for simple diffusion. Facilitated diffusion is a carrier mediated transport. So basically the difference between these two, simple diffusion occurs through the lipid bilayer or through the channels. Facilitated diffusion is carried out by the carrier. Let's see the various aspects of these two and also the examples. Let's start with the simple diffusion. Already we have mentioned the simple diffusion has two ways, either through the lipid bilayer, through the watery channels, or the water soluble substances going through the channels. There is a Fick's first law for simple diffusion. Fick's first law for simple diffusion. It delineates the factors influencing the rate of simple diffusion. It says J, that is flux, the rate of simple diffusion, is equal to minus D into A into delta C upon D. J is the rate of simple diffusion, is equal to these are the factors to which it is proportional. Minus sign indicates downhill transport. Capital D is diffusion coefficient. For each substance, there is a diffusion coefficient which will determine the speed of diffusion of that substance. A is surface area of the membrane. Delta C, concentration gradient. You can say C1 minus C2. It's going to go from high to low concentration. So there are two concentrations on either side of the membrane, C1 and C2, and larger the concentration gradient, faster will be the rate of diffusion, as you can see directly proportional. And then D, small d, thickness of the membrane or diffusion distance. Diffusion distance. So, if I were to ask, the rate of simple diffusion is inversely related to thickness of the membrane or diffusion distance. The other factors, rate of diffusion will be directly proportional. Alright, now let us discuss these factors in detail with the suitable examples. So, the next point is factors influencing the rate of simple diffusion. Examples are important, so I would suggest you to look at the examples carefully. Okay, there are 8 factors to which the rate is proportional. Let's look at those factors one by one. First 2 factors are easy to understand. We have already said that simple diffusion can occur in 2 ways, through the lipid bilayer and through the channels. So we have 2 clear-cut factors. Lipid solubility, yes, lipid soluble substances, they go through the lipid bilayer, and the rate of simple diffusion will depend on the lipid solubility of the substance. Greater the lipid solubility, faster will be the rate of simple diffusion of that substance. But look at the example. CO2 is almost 20 times more lipid soluble compared to oxygen, and hence the rate of carbon dioxide diffusion will be much faster as compared to the rate of oxygen diffusion. Okay, carbon dioxide, let's say lungs, carbon dioxide diffuses from the pulmonary blood vessel into the alveolus, and oxygen from the alveolus goes into the pulmonary blood. Carbon dioxide will reach equilibrium very rapidly. It diffuses through the membrane very rapidly. The reason is this. First factor, then second, number of channels available in the membrane. The water soluble substances, they are diffusing through the channels, and therefore naturally more the number of channels available, greater will be the rate of diffusion. Now look at the example. Number of potassium channels is higher as compared to the number of sodium channels in a cell membrane, and therefore potassium diffuses faster as compared to the diffusion of sodium. Please keep this in mind. Potassium diffusion rate is faster as compared to the sodium diffusion because potassium has got more number of channels in the membrane. I am going to discuss this in detail. Let me just arouse the interest. Let me just ask you 3 questions, one after the other. First question. Most diffusible ion for a cell membrane, through a cell membrane is? That's the first question. The options are sodium, potassium, calcium, chloride. Hold your thoughts. In the end, we are going to discuss this MCQ in the end of this section. Second MCQ. Most diffusible ion in the case of an excitable cell is? First question was most diffusible ion for any cell, for any membrane, would be which ion? Sodium, potassium, calcium, chloride. Second question, most diffusible ion in the case of an excitable cell membrane, nerve or muscle membrane. Take same 4 options, sodium, potassium, calcium, chloride. Third question, most diffusible ion in the case of an exciting membrane. When the membrane is exciting, it's about to excite, its excitation has started, then which will be the most diffusible ion, or which ion will diffuse faster? And the same 4 options, sodium, potassium, calcium, chloride. We are going to answer these 3 questions in next 5 to 10 minutes. Okay. Third factor that influences the rate of simple diffusion. Temperature. Well, we will just add one point here. I said facilitated diffusion occurs by virtue of carrier. It's a carrier mediated transport. Simple diffusion occurs due to the kinetic energy of the particles. Kinetic energy is what drives the simple diffusion. Okay, so let that be clear. Simple diffusion is driven by kinetic energy. Facilitated is by the carrier. So, coming back. Now it's obvious. To this factor also, the rate of simple diffusion will be directly proportional. Higher the temperature, greater will be the kinetic energy, and therefore greater will be the rate of simple diffusion. Right. Next, surface area of the membrane. Surface area of the membrane. Okay. Again, directly proportional. So far we have discussed 4 factors, and for all those 4, they were directly influencing the rate of directly proportional was the rate of simple diffusion. Example over here, surface area. Let's say emphysema. If you have heard this condition, emphysema, destruction of the alveolar septa and alveoli coalesce to form larger cavities. But then surface area for diffusion decreases in the condition of emphysema. The condition of lungs, over distension of the alveoli with air and destruction of the alveolar septa. So because the surface area for gas diffusion decreases, the rate of gas diffusion will decrease. More the surface area, more will be the rate of diffusion. Less the surface area, as in this case, the rate of diffusion will decrease. And that's one problem in emphysema. Oxygen diffusion rate decreases. Carbon dioxide diffusion rate also decreases. Right. So far we have seen 4 factors. Before we proceed further, I told you there are about 8-9 factors. We will see those factors one by one. Before we proceed further, I am just going to ask you one small little question. I want you to think about it and we will discuss the answer later on. We said just now that temperature influences the rate of diffusion directly. But there is a condition called as multiple sclerosis. It's a demyelinating disease of the neuron. Demyelination of the neurons. The neurons are denuded and there is no myelin sheath, or for the large part of the neuron there is no myelin sheath. So demyelination occurs. Now it has been observed in those cases that hot bath, look, basically in multiple sclerosis, the symptoms are related to the conduction defects, action potential conduction, impulse conduction is affected. Sometimes it is blocked. No proper conduction of impulses. Hot bath has been shown to worsen the symptoms of multiple sclerosis. The symptoms become worse. They have to be sometimes given cold packs, decrease the temperature so that the symptoms are relieved. Now, look, on one hand we are saying greater the temperature, greater is the diffusion. Action potential through a neuron, through a nerve fiber is because of the diffusion of sodium, isn't it? Depolarization, excitation of the membrane is because of the diffusion of ions, diffusion of sodium. So increased temperature should make the diffusion better by our own principle. But in multiple sclerosis, it has been observed that increased temperature, that is hot bath, the symptoms of conduction block become worse. Diffusion decreases or something like that. What's the reason for that? Think about this. We will come to the answer in the relevant section when we discuss the nerve. Okay. Next 2 factors are those factors to which the rate is inversely related. Only 2 factors, so it's easy to remember, 5 and 6, 2 factors, the rate of diffusion is inversely related. One we have already seen, thickness of the membrane through which diffusion is taking place. We can call it diffusion distance as well. So, the rate of simple diffusion is inversely proportional to thickness of the membrane or diffusion distance. Thicker the membrane, more time will it take to travel from one side to, more time will be taken by the substance to travel from one side to the other side of the membrane. So that's very logical to understand. Now, what is the example that we can give? Pulmonary edema. You know, here is a condition in which the thickness of the respiratory membrane increases and therefore the rate of gas diffusion. Look, here is an alveolus, here is a pulmonary blood vessel, and oxygen diffuses through this respiratory membrane from alveolus to the blood. In pulmonary edema, thickness of the respiratory membrane increases and therefore gas diffusion, oxygen carbon dioxide diffusion will decrease. Yeah, so inversely proportional. Another factor, size of the diffusing substance or molecular weight of the diffusing substance or radius or diameter of the diffusing substance. This also is quite easy to understand. Larger the particle, slower will it diffuse. You know, in humans also, more the weight, the velocity might become slower. That's not the rule. So just forget about what I said just now. But here, definitely, larger the particle, then slower will be the rate of diffusion. Alright. Now listen to the example. We are comparing sodium and potassium. And both these ions are hydrated in the body. In the body, they are in a hydrated status. Hydrated sodium has got a diameter of 2.2 and hydrated potassium ion has got a diameter of 2.0 nanometer. So which particle will move faster? Smaller the particle, faster will it diffuse. Okay, and therefore potassium will diffuse faster as compared to sodium. So I have told you second reason why potassium diffuses faster as compared to sodium. First reason was more number of channels for potassium as compared to sodium. And the second one is this, the size. Okay. Now last three factors are very similar to understand and remember. So we'll discuss them together. Concentration gradient. Concentration gradient. Okay, last three factors are the ones for which diffusion rate is directly proportional. So larger the concentration gradient, faster will be the rate of diffusion. Directly proportion. Look at the cell and sodium and potassium concentrations. Sodium 141 milliequivalents per liter on the outside, 14 milliequivalents per liter on the inside. This is a concentration gradient for sodium. Potassium, now look at the potassium. 145 milliequivalents per liter on the inside and only 4 milliequivalents per liter on the outside. Come on, tell me whose concentration gradient is wider? Look at the values. Isn't it obvious? Potassium. Potassium has a wider concentration gradient, greater concentration gradient, and therefore potassium diffusion rate will be faster as compared to sodium. So I've told you third reason, three reasons in all why potassium diffuses faster as compared to sodium. And therefore now let's come to the MCQs or questions that I asked a few minutes ago. The most diffusible ion is for any cell. Answer is potassium. Let me write down. The rate of potassium diffusion is 50 to 100 times greater as compared to the rate of diffusion of sodium. Potassium diffuses 50 to 100 times faster as compared to sodium. Fine. Potassium diffusion rate is faster for any cell. Second question was most diffusible ion for an excitable cell and its membrane. Answer will be again potassium. Look, whether it's an RBC or a hepatocyte, those are non-excitable, or an epithelial cell, these are non-excitable cells. Nerve and muscle are the excitable cells. Their membranes excite. Even for those cells, potassium will be the most diffusible ion. Fastest diffusion will be that of potassium. So second MCQ answer was potassium. Now coming to the third one. Most diffusible ion for an exciting membrane. Look, I told you excitable cell and its membrane. That time we did assume that the cell is resting. It's not exciting as of now. In that case, potassium diffusion rate will be faster. But the third MCQ was, when the membrane is exciting, then which ion will diffuse faster, sodium or potassium? Now this is a trick question, and for that the answer was sodium. When the membrane is stimulated, nerve membrane, muscle membrane, when it is stimulated and it begins to excite, sodium diffusion takes precedence over potassium diffusion. Sodium coming in happens at a much faster rate and as compared to potassium going out, and therefore the membrane excites. So remember, it's a play of words actually. It's a play of words. I mean, most diffusible ion for a cell, answer potassium. Most diffusible ion for an excitable cell, answer again potassium. So for any cell, potassium diffuses faster. But most diffusible ion or faster diffusion is that of which ion? For an exciting membrane. The membrane has begun to excite. That time sodium takes precedence over potassium, and therefore the answer, look, this is just a play of words, and just to answer the MCQ we need to look at the words carefully sometimes. Right, that was the next factor, concentration gradient. But just remember, potassium diffusion rate is faster. Anyways, we are not considering the other ions as of now, chloride, calcium, etc., because their movement is much less as compared to these two ions. Remember, for most of the times these two ions are making the maximum movements, and therefore in the membrane, membrane of every cell, we have sodium potassium pump, which will rearrange the concentrations of sodium and potassium after they have moved. Therefore we always compare sodium and potassium only. We don't consider the other ions, I mean to say. Okay, pressure gradient, next factor. Pressure gradient, it's very similar to concentration gradient. Pressure gradient is for the gases. Gases diffuse from high pressure to low pressure. So pressure gradient is responsible. Take the example. What is the importance of giving hyperbaric oxygen? Let's say in carbon monoxide poisoning. The reason, the basis, the logic is that when you give hyperbaric oxygen, oxygen under high pressure, it reaches alveoli with a high pressure, because you are giving it under high pressure. So partial pressure of oxygen in the alveolus will increase dramatically, tremendously, and therefore its pressure gradient from alveolus to the blood will also widen. I mean, just imagine that in the blood the PO2 was 40, and normally in the PO2 in the alveolus was, let's say, 104. 40 in the venous blood, it has come to the pulmonary circulation now. This was one pressure gradient normally, and now I give hyperbaric oxygen, and instead of 104 in the alveolus, it becomes 304. Let's just some hypothetical figure. So 304 to 40, this side of the membrane, the pressure gradient has widened, increased. The gas will diffuse faster, the oxygen will diffuse faster. Just an example. Okay, last factor is electrochemical gradient or electrical gradient. Look, the ions, they not only move because of the concentration gradient, they also move because of the electrical gradient or electrochemical gradient. Remember this, it's a very important factor in the diffusion of substances through the epithelial cells. Why I mentioned epithelial cells is because epithelial cells are they are said to have polar cells with charges lined up on one side of the cell, and in that case the transport of ions is driven by electrochemical gradients. So this is very important factor. We will consider it later when we discuss the kidney and renal tubules and transport of substances for reabsorption. But to have it a simple example in this, we have discussed it at the start of the cell that there are excess anions inside the cell. The proteins and phosphate, they make excess anions inside the cell and relatively less cations. And there are more cations in the ECF. So a cation like sodium or even potassium for that matter will diffuse from high number of positive charges to relatively lower number of positive charges or relatively higher number of negative charges. Okay, so sodium moving from high number of positive charges to low number of positive charges, this is the diffusion driven by electrical gradient or electrochemical gradient. It's very important, and we will have the concepts based on this particular aspect, electrochemical gradient, when we discuss the action potential or equilibrium potential and all the other things. So please keep this in mind, that ions not only diffuse by concentration gradient, but they also diffuse by the electrical gradient or electrochemical gradient, and wider the electrochemical gradient, greater will be the rate of diffusion. Right. These were the factors influencing the rate of simple diffusion. Second type of diffusion is facilitated diffusion. Facilitated diffusion. Let's understand a few basic things about it. Facilitated diffusion is a carrier mediated transport. Carrier drives this particular transport, and again net facilitated diffusion is a downhill transport. So, and it's a passive transport. Passive transport, that means no ATP needed. So how does the transport occur? There will be a transformational change, configurational change in the carrier, and that will cause the substance to move from one side of the membrane to the other side. Let's see the characteristics of the facilitated diffusion, and then we will see the example. First characteristic is saturability. Saturability. The facilitated diffusion shows saturation kinetics. I mean, if you have read some biochemistry, they will tell you it follows Michaelis-Menten kinetics. Saturation kinetics or Michaelis-Menten kinetics. Carrier gets saturated, and therefore beyond a certain point it will not be able to increase the rate of facilitated diffusion. And to understand this, we will have a comparative graph of simple versus facilitated diffusion. On this axis there is rate of transport or rate of diffusion, and over here on the horizontal axis concentration of the substance on one side of the membrane. Let's take some arbitrary units here to understand. 10, 50, 100, 500, let's say millimoles per liter per, yeah. What I'm trying to say in this graph is how the concentration of substance will be increased on one side of the membrane, and that means, of course, concentration gradient will also increase. See the graph for simple diffusion. Straight line, linear. It's a linear graph. So you go on increasing the concentration of the substance on one side of the membrane, the rate of simple diffusion will go on increasing. Infinity. I mean, here is the simple diffusion, and this is a concentration gradient, 100:10. Diffusion will occur 500:10. Concentration was increased on one side. The rate of simple diffusion will increase. 1000:10. Concentration increased further on one side of the membrane. The rate of simple diffusion will further increase, and it will go on increasing. If you increase the concentration gradient, if you increase the concentration on one side of the membrane, the rate of simple diffusion will go on increasing. Oxygen. If you give more and more oxygen, if you give 100% oxygen, see, the example is, we are now inspiring atmospheric air, and oxygen in this air is 20%. Instead, if you give 100% oxygen, that means you have increased the oxygen concentration into the alveolus by 5 times. Oxygen diffusion rate from alveolus to the blood will also increase proportionately. So simple diffusion has this advantage. You increase the concentration, the rate of simple diffusion will go on increasing. Now coming to the facilitated diffusion, look, initially the rate of facilitated diffusion will also increase. See, we have increased the concentration on one side of the membrane, and we see the graph is rising, rate of diffusion is increasing. But beyond a certain point, beyond a certain concentration, let's say here, beyond this, now you increase the concentration further from 50 to 85 to 100 and so on. The rate of diffusion has not gone up, it's flattened. This is the maximum, and it stays at maximum. You see this line, flat, plateau, it says the rate increased, reached maximum, and it reaches maximum. Now why does that happen? Is because of the saturation. At this point, carrier got saturated. When you increase the concentration on one side of the membrane, more and more molecules of that substance will attach to the carrier. So carrier can deliver the substance faster and faster. But one time will come when carrier will be completely saturated. If you were increasing the concentration of that substance on one side, and more were attaching, but at one point the carrier will get saturated. So beyond that point, the rate cannot increase any further. That will be the maximum rate at which the carrier will continue to deliver the substance. Yeah, so you can see it remained flat. It remained maximum at that level, and it remained so. So you can see comparison of these two graphs. Let me just write here, this is for simple diffusion. This is for facilitated diffusion. And of course, this maximum velocity or rate with which the transport happens, it's called as Vmax, this flat line. So that's the comparison. So you must remember 2 points from this graph. One is this Vmax characteristic, saturability. And second point is, up to a certain concentration, now this is important, please remember this, up to a certain concentration, the rate of facilitated diffusion will be faster as compared to rate of simple diffusion. See, up to this point, the graph is steeper for facilitated diffusion compared to the simple diffusion. And then beyond that point, you can see the graph has increased further for simple diffusion. but not for facilitated. Well, this is just a hypothetical comparison. Let me end this point by saying this is hypothetical comparison, because we don't have an example in the body where same substance is being carried by simple diffusion and facilitated diffusion so that we could compare both. No, it's not the case. So therefore, this is just a hypothetical scenario or comparison. Second characteristic of facilitated diffusion is specificity. Think about simple diffusion. There was no specificity. Any lipid soluble substance can diffuse through the lipid bilayer. No specificity. But facilitated diffusion means if the substance has to be transported, it has to have specific carrier in the membrane to carry that particular substance. And the third one is inhibition. Third characteristic of facilitated diffusion, inhibition. You know, simple diffusion cannot be inhibited or stopped. As long as the concentration gradient is there and it allows the diffusion, diffusion will continue. It can't be inhibited. Facilitated diffusion can be inhibited, can be stopped. And it is of two ways, you know, enzymatic inhibition and non-enzymatic inhibition. If you inhibit the carrier, the transport will stop. Facilitated diffusion can be stopped. In fact, I can tell you the example. Glucose transport occurs by facilitated diffusion. And there is a poison which inhibits the glucose carrier and stops the transport. It's fluorescein. Some authors also write it as fluoretin. So fluorescein or fluoretin, it's a specific poison for glucose transport. We are going to see the example of facilitated diffusion, that's glucose transport. How is it? What is the substance that inhibits it? Please remember this. I will just add one more point and then we'll see the glucose transport after that. Look, if you remember, we discussed the carriers in the membrane. And I had told you that carrier is responsible for two types of transports, facilitated diffusion and remember which was the second one? Secondary active transport. These two are carrier mediated transports, okay? Both are carried out by the carrier. And I had also mentioned that in the case of facilitated diffusion, I have mentioned this just now, the substance always goes from high to low concentration. I mean, net diffusion occurs from high to low concentration. In the case of secondary active transport, it goes from low to high concentration. My point is, and I'm going to ask you the question and take the answer also, or answer the question. Carriers can carry the substances from low to high concentration also. Look, in the case of secondary active transport, the carrier carries the substance from low to high concentration. Even in facilitated diffusion, the substance goes from high to low as well as low to high, and net transport is high to low in the case of facilitated diffusion. So my question to you, and of course I'm going to answer the question, is why is it that the carrier in the case of facilitated diffusion compulsorily carries the substance from high to low concentration only when it can carry also from low to high? The answer is, well, whenever I ask this question, many students, they just simply say, in the case of secondary active, there was ATP used, therefore. No. That's not the correct answer. Look, you have to know the principle of facilitated diffusion in order to answer this question. In the case of facilitated diffusion, the principle on which the carrier is based and the transport is based is high concentration is equal to high affinity for the carrier and low concentration is equal to low affinity Okay, high concentration is equal to high affinity, low concentration is equal to low affinity. That's the principle on which facilitated diffusion and its carrier is based. So therefore, wherever there is high concentration, the substance will bind, and wherever there is low concentration, it will be delivered. And therefore, net facilitated diffusion always occurs from high to low. It can go low to high, remember, but net occurs from high to low concentration. And finally, let's see the example of facilitated diffusion. Glucose transport and glucose entry into most cells of the body occurs by facilitated diffusion, except in the GIT and kidney where glucose enters the cells by secondary active transport. We will see that later. And the carrier is called as GLUT. So remember, glucose entry in almost all cells. Let's see the types of GLUTs. GLUT1 is called as constitutively expressed glucose transporter, and it is ubiquitous, omnipresent. So this is present everywhere in the body. You mention the cell and generally it will have GLUT1. So RBCs, etc. Most cells have GLUT1. Ubiquitous, omnipresent. GLUT2, very important points to be remembered. Pancreatic beta cell has GLUT2. So glucose entry into the pancreatic beta cell occurs by GLUT2. And one more, which we have actually discussed it previously. Glucose exit from the cells whenever it has to happen. Normally glucose enters and gets metabolized. But in the case of GIT and kidney, glucose leaves the cell also to enter the blood. Or in glycogenolysis, glycogen breakdown happens and that glucose which is formed, it comes out of the cell into the blood. So glucose exit occurs by using GLUT2. GLUT3, this question was asked in some previous exams. Neuronal tissue predominantly has which GLUT? 1, 2, 3, 4. The answer was 3. Predominantly, okay. So neurons predominantly have GLUT3. They do have GLUT1 as well, but predominant transport of glucose into the neurons occurs by GLUT3. And then of course our favorite one, GLUT4. This you must be knowing very well. It's called as insulin dependent glucose transporter. Right? And therefore this is present at two places: adipocytes, fat cells, and the muscle. The skeletal muscle and the adipocytes, these two are the tissues which are dependent on insulin for glucose entry, because these two tissues and their cells have GLUT4, which is insulin dependent glucose transporter. GLUT5, this question has come very often. Spermatozoa and their membrane has which type of GLUT for glucose entry? 1, 2, 3 or 5? Answer is 5. Fructose transport occurs by using GLUT5. And you know spermatozoa metabolize fructose. So they have GLUT5. That was the facilitated diffusion. Finally, I will ask you one question. We'll discuss something about diffusion and then move on to the active transport. The question was, aspirin, you know the drug aspirin, NSAID, it enters the parietal cell of the stomach by what type of diffusion? And the options are simple diffusion, facilitated diffusion, non-ionic diffusion, or translational diffusion. Are you answering that question? Well, the answer was non-ionic diffusion. Non-ionic diffusion. So a special type that I wanted to mention in the end, non-ionic diffusion. Well, also called as diffusion trapping. It's also called as diffusion trapping. First thing, which substances are transported like this? Weak acids or weak bases. Their transport is described as non-ionic diffusion. What is it? Okay. Their non-ionic forms are more diffusible. So you need to remember this one particular basic fact. This will be required in the pharmacology later on. Non-ionic forms are more diffusible. They can diffuse through the membrane. They are more diffusible. Ionic forms are less diffusible. Okay. So look at this. Non-ionic form of a particular acid. It is more diffusible, so it diffuses through the membrane, goes to the other side of the membrane and acquires an ion. This is just a diagrammatic representation. It acquires an ion. Now this has become ionic form, and I said already ionic forms are less diffusible. So this now cannot diffuse back through the same membrane. That means what? That means it has got trapped on the other side. It diffused readily, it went to the other side, acquired an ion, and now it can't go back. It is trapped on the other side. So this is also called as diffusion trapping. Let me give you the examples. Aspirin entry into the parietal cell of the stomach. That's see, aspirin is a weak acid, acetyl salicylic acid, and when it is taken by mouth, it enters the parietal cell and acquires an ion. Now it goes into ionic form, so it cannot come out of the cell. It cannot diffuse back. So it's trapped. It is trapped in the parietal cell. And this is one of the reasons of ulcerogenicity which is shown by the aspirin. Second, ammonia and its buffering in the collecting duct in the kidney. Remember, ammonia is the buffer for chronic respiratory acidosis. Respiratory acidosis and alkalosis will be compensated by the kidneys. You must be aware of that, or we will discuss it later. Now look at the ammonia. One molecule of glutamine produces two ammonia molecules. So this is a cell which is producing this ammonia. Cell in the collecting duct. Now this ammonia comes out into the lumen and it acquires an H+. There was an H+ to be eliminated into the urine. So now this ammonia will acquire this H+ to become NH4+, ammonium. Now this is ionic form. So it can't diffuse back. And therefore it will be excreted into the urine now. And that's how it will eliminate this H+ into the urine. That is diffusion trapping or non-ionic diffusion. That was about the transport by diffusion, and we saw various examples. Finally, we will talk about the active transport, and then we will end with sodium potassium pump. So let's active transport is it uses ATP and it is an uphill transport. It's an uphill transport. That is, substance is transported from low to high concentration. Uphill transport. So whenever a substance goes from low to high concentration, it is some kind of an active transport. and it uses ATP. So now, based on how it uses ATP, there are two types of active transports. One, the primary active transport, when ATP is used directly. ATP is used directly, then it will be called as primary active transport. Example, our favorite sodium potassium pump. Sodium is bound, potassium is bound, ATP is bound. ATP is broken down and then sodium and potassium will be transported from their low to their high concentrations. So ATP was used directly, I mean to the same protein, ATP was bound and broken. When ATP is used indirectly, then it will be called as secondary active transport. When ATP is used indirectly. Now what is the meaning of this? Indirect use of ATP. What's the meaning? Before I tell you this indirect use of ATP, let me just add one or two lines here. In the case of secondary active, there are two types. Remember we discussed carrier mediated transport. Facilitated diffusion was called as uniport and this was called as symport and antiport. Two substances are going to be transported by a carrier. So first of all, remember secondary active transport uses carrier. It will carry two substances together simultaneously. And when those two substances, they go in the same direction, they are going in the same direction, it will be called as symport or co-transport. And if they are going opposite to each other, then it will be called as antiport or counter transport, or even it's called as exchange. Okay, so let's look at these examples one by one. Symport. Glucose is transported into the cells of GIT and kidney by symport or co-transport. Okay, glucose entry in other cells of the body by facilitated diffusion, and here it's by secondary active transport. Glucose will go from low to high concentration. Remember, that's why it's called as secondary active transport. And the transport protein is called as SGLT, sodium glucose transporter. Okay. SGLT1 is predominantly in the GIT and it carries 2 sodium is to 1 glucose. You must be aware of this by now. We are talking of two substances transported simultaneously, sodium and glucose. But remember, sodium goes from its high to its low concentration and glucose is going from its low to its high concentration. Please remember this. I am going to come to that in some time. So SGLT1, sodium glucose transporter, 2 sodium to 1 glucose it transports, and it's mainly in the GIT, and its defect results in glucose galactose malabsorption. Whereas SGLT2 and SGLT3 are predominantly in the kidney and they carry 1 sodium to 1 glucose. Please remember again, they both are entering the cell, but so they are both are them going in the same direction, but sodium is going from its high to its low concentration and glucose is going from its low to its high concentration. So for the glucose, it is the active transport going from low to high concentration. That was symport, and the other one is antiport or counter transport or exchange. So if sodium is going in one direction, then the other substance goes in the opposite direction to that of sodium. This is called as antiport or counter transport or exchange. I will explain this mechanism in next 3-4 minutes, how this happens, two substances transported. But antiport is sodium and the other substance are going in the opposite directions. Sodium is going to from its high to its low concentration. The other substance is going from its low to its high concentration. Example, sodium calcium exchanger in the heart, NCX in the heart. It carries 3 sodium is to 1 calcium. The stoichiometry of sodium calcium exchanger is 3 sodium is to 1 calcium, and therefore how it will happen? 3 sodium are going in and 1 calcium ion is coming out. That is the exchange. That is the counter transport. Or the other ones like chloride bicarbonate exchanger in the RBC, the band 3 protein it is called. Right. The final point which I want to make here is all the secondary active transports are absolutely dependent. They are driven and they are absolutely dependent on the primary active transport, and mostly it's sodium potassium pump, the primary active transport. Therefore, let me write it here. They are dependent on the sodium potassium pump. If the sodium potassium pump fails, all the secondary active transports will also fail. Okay, please, I want to repeat this particular line again. They are all absolutely dependent on sodium potassium pump. If the sodium potassium pump fails, all the secondary active transports will also fail. They will stop. They will not happen. The carrier will not be able to carry that transport. Why? Let's see here. These secondary active transports absolutely dependent on sodium potassium pump, right? See how they happen. I want to describe you to that so you will understand why they are absolutely dependent on sodium potassium pump. Sodium potassium pump, the primary active transporter, it does what? It creates and widens the concentration gradient for sodium. Look, sodium potassium pump is doing what? It is throwing the sodium out of the cell. That means sodium will be higher on the outside and lower on the inside of the cell, isn't it? By the action of the sodium potassium pump, the pump has thrown out the sodium. So sodium will be high on the outside and remain low on the inside. So sodium potassium pump creates and widens the concentration gradient for sodium. Now I am making a critical point. In this concentration gradient, there is a potential energy, there is a pull by which sodium will be now driven inward from high to low concentration. Okay? This potential energy, the potential energy stored in the concentration gradient, the potential energy stored in the concentration gradient, that pull created by the concentration gradient will be used by the carrier to carry two substances at a time. Sodium will be taken from high to low concentration, and the other substance will take advantage of that and will go from its low to its high concentration. Yeah. So carrier is going to use the potential energy stored in the sodium's concentration gradient, and it will cause sodium to move from high to low concentration. But at the same time, another substance will take advantage of this, and another substance, second substance, will move from its low to its high concentration. So please remember, you might be thinking all along that secondary active transport, oh, it's active, means it must be utilizing ATP. No, the carrier does not use ATP. ATP is used by sodium potassium pump, which is the ATPase. But that use of ATP creates a concentration gradient for sodium. And that concentration gradient now drives the sodium from high to low concentration. And that drive is utilized by the carrier to carry another substance from low to high concentration. So secondary active transport will be for that another substance, right? And if the second substance goes along with the sodium in the same direction, if it is entering the cell, it will be called as symport. If the second substance is leaving the cell, because see, sodium is going to come in by high to low concentration. If the second substance is leaving the cell at the same time, it's going opposite. It will be called as antiport or counter transport or exchange, which we have already mentioned. The examples we have seen already. But the point was, let me just finish this flowchart. Potential energy stored in the concentration gradient is used by the carrier to carry one, sodium from high to low, and second substance from low to high. So that second substance is being carried in the form of secondary active transport. Right, so we have seen the examples of this already, the types and the examples. But my point was, if the sodium potassium pump fails, then all the secondary active transports will also fail. Please remember this. I mean, for instance, there is some drug which binds to the sodium potassium pump and inhibits the sodium potassium pump. The drug or poison that binds and inhibits the sodium potassium pump, in that case, the secondary active transport based on that will also be inhibited. So that's the active transport and its types. Finally, we will also discuss sodium potassium pump. I had kept separate time for sodium potassium pump because it's a very important topic to be understood. So here comes the final part of this chapter, the sodium potassium pump. I had deliberately kept this as a separate part, although it's a primary active transport and we have described primary active transport before this. But most of the processes in the body are dependent on sodium potassium pump, and more than half the pathophysiological processes, also disease processes, are basically, I mean, you can put the blame on sodium potassium pump and its failure, resulting in those pathophysiologies. So a very important aspect of the transports. Let's understand this. Just before this, when we discussed secondary active transport, we said that secondary active transports are absolutely dependent on sodium potassium pump, which is a primary active transport. Sodium potassium pump breaks down the ATP, throws the sodium out of the cell, and that creates the concentration gradient for sodium. Now that concentration gradient has a certain potential energy, certain pull, and that pull is utilized by the carrier to carry out the secondary active transport of, the sodium will be pulled into the cell from high to low concentration, and at the same time, one more substance will be transported. If it goes into the cell, that means it's going along with sodium in the same direction, so it will be called as co-transport or symport. And the second substance goes opposite to sodium, then it's called as antipode. So we've described all this. Now let us understand sodium potassium pump, a very, very important discovery. In fact, the discoverer was given Nobel Prize in 1998. Jens Skou discovered the sodium potassium pump. I had to mention this because it was considered a very important discovery of the century. Right. Sodium potassium pump is present in all the membranes. All the cell membranes have sodium potassium pump. It's not as if it's there only in nerve or muscle or this or that. All cell membranes have sodium potassium pump. You take an RBC, it has sodium potassium pump in nerve, muscle, cardiac tissue, anywhere, everywhere. And it is active all the times. At most places, this is active all the time. I mean, it's not as if something happens, impulse goes and then it becomes active. No. It's continuously performing its job of sodium and potassium transport. Why does it do that continuously? We will see that in some time. Basically, the sodium potassium pump has 3 subunits. Now, I know I'm making a debatable statement here because most of you have read 2 subunits, alpha and beta. Well, third subunit has been discovered recently. And as I mentioned previously, that third subunit, gamma subunit, belongs to FXYD category of proteins. So, sodium potassium pump has 3 subunits, alpha, beta, gamma. Look, the alpha subunit is catalytic. Means all the binding and ATP breakdown and the transport of ions, it happens on the alpha subunit. Beta and gamma are only supportive. They target the pump to the membrane, means it's synthesized, is formed inside the cell and then targeted to the membrane, and in a way they regulate the turnover of the sodium potassium pump. Right, so alpha subunit is what we are interested in. Alpha subunit has 5 binding sites. 3 on the intracellular aspect of the pump and 2 on the extracellular aspect of the pump. 1, 2, 3, 4 and 5. I have written the numbers based on the binding of those particular substances. So let's see. Number 1 is sodium binding site, and it's on the inner aspect of the pump, facing the intracellular aspect. Number 2 is potassium binding site, which is facing the extracellular aspect of the pump. 3, ATP binding site. Number 4 is phosphorylation site. And number 5 is Obein binding site. So these are the 5 binding sites on the alpha subunit. Let us see the activity of the sodium potassium pump. I mean, what does it achieve? I mean, what does it perform as a function? Look, it is continuously causing the sodium and potassium ions to move from their low to their high concentrations. Potassium is low in the ECF and high intracellularly. So potassium comes from outside to inside, low to high. And similarly, sodium is low on the inside and high in the ECF. So sodium is taken out. And how does it happen? 3 sodium ions bind to the internal aspect of the pump. Well, let me just add one more point. This pump is also called as E1, E2 pump. E1, E2 is, these are the 2 conformational states of the pump. In the E1 state, it binds the sodium, binds the ATP, and then sodium is thrown out. And then in the E2 state, it will bind the potassium. 2 potassium ions from the outside are taken into the cell, are transported into the cell. So a primary active transport causing 2 substances to be transported in the opposite direction, but it is not antipode. Many students ask this question, why can't it be considered antipode exchange? No. Both the substances are going from their low to their high concentrations. Remember, in the secondary active transport, as in antipode or exchange, one was sodium, which was going from high to low, and only the other substance was going low to high. Here both substances are going low to high concentration. Right. So 3 sodium ions from inside to outside, 2 potassium ions from outside to inside. 3 sodium ions from inside to outside, 2 potassium ions from outside to inside. This is being done continuously everywhere in the body. Okay. And therefore, I had mentioned this before, that we always talk about sodium and potassium ion concentrations. We don't discuss any other ion concentrations. The reason being, the other ions hardly move through the membrane, and therefore their concentrations hardly get disturbed, if at all. It's this sodium and potassium ion concentrations which continue to get disturbed all the time, and therefore we have something like a sodium potassium pump, which is going to cause redistribution of those concentrations. Right. Now with that in mind, let us see the functions of the pump. Well, just before that, let me just add one point here, pharmacologically important. Potassium and obein derivatives as in digitalis, the cardiac glycosides. The digitalis and potassium, they compete for the binding sites which is on the outside of the membrane. Potassium and the digitalis. So they are competing for those binding sites. Now I have mentioned this before, that digitalis binds to the sodium potassium pump and it inhibits the sodium potassium pump. If it inhibits the sodium potassium pump, what will happen? The concentration of sodium ion cannot be established. Concentration gradient for sodium cannot be established if sodium is not being thrown out. And if the sodium concentration gradient cannot get established, the secondary active transports which are based on that, they will also not happen. And one secondary active transport is NCX. It brings the sodium in and sends the calcium out. That will not happen. Because digitalis is bound to the sodium potassium pump, it inhibited the sodium potassium pump, therefore sodium could not go out, therefore there was no sodium concentration gradient from outside to inside. So the carriers for the secondary active transport cannot work, and one such carrier is sodium calcium exchanger. Remember we said it brings 3 sodium in and sends 1 calcium out. That cannot happen. Which means what? Which means calcium cannot be sent out by that carrier. Calcium stays on inside the cell. Calcium concentration in the cell increases. If that increases in the myocardial fiber will contract strongly. That's the mechanism of action of digitalis or cardiac glycosides. So with that in mind, just I had to add this particular information because potassium we discussed binding on the outer side, where we have mentioned the 5th binding site, the obein binding site. But basically they are competing for those binding sites, potassium and obein. So remember, therefore hypokalemia results in digitalis toxicity. Hypokalemia, if there is low potassium in the plasma, then it will result in digitalis toxicity, because if there is low potassium means the competition for digitalis for the binding sites will be less. There is less potassium to compete with digitalis, and therefore digitalis binding will be excessive, its action will be excessive, it will result in toxicity. Or in digitalis toxicity, we can administer potassium. We will create the competition for the digitalis to bind with the sodium potassium pump. Right. Let's see the functions of the sodium potassium pump with all of this. First is sodium potassium pump opposes the equilibrium. It's the opposer of the equilibrium. It's not a contributor to the equilibrium. It's an opposer to the equilibrium. It does not allow equilibrium to happen. And see how. We have been saying from the start of this chapter that intracellularly there are lots of anions, and in the form of proteins and phosphate. So excess anions in the cell, inside the cell. And to have the equilibrium, the law of electroneutrality or Gibbs-Donan equilibrium, equal number of cations should be pulled in. There should be an equal number of cations and anions. So the cation that gets pulled in to the cell is the sodium. For what? To achieve that electroneutrality or to achieve the equilibrium. So we have also already seen this, that sodium is continuously leaking into all the cells of the body, everywhere, continuously. What for? For this reason. But then imagine that sodium leaking into all the cells continuously, sodium comes in and stays. Sodium concentration begins to increase inside the cell. What will happen? Sodium is osmotically active. So sodium concentration increases inside the cell means what? Sodium will be followed by water. So water will enter all the cells and the cells will burst, all of them. So remember, sodium is attempting to reach the equilibrium, but that is opposed by sodium potassium pump. Okay. How? Look, sodium is leaking in to all the cells to attempt that equilibrium, but sodium potassium pump will throw the sodium out. Whatever sodium is coming in, sodium potassium pump will throw it out. It will not allow the sodium to rise inside the cell intracellularly. Else there will be water entering into the cell and cells will swell. So that's the job of sodium potassium pump. It will oppose that equilibrium from happening. It will not allow it to happen. Second function is a fallout of the first one only. It regulates the cell volume. Sodium potassium pump regulates the cell volume. By keeping the sodium low inside the cell, by throwing the sodium out, cell volume is regulated, because if the sodium stays more inside the cell, water will come in, cell volume will increase. That's another function. Third function is sodium potassium pump is involved in the process of recharging of the excitable cells. See, slowly you must have started understanding why sodium potassium pump is continuously active everywhere in the body. What is the need for that? In fact, many students, without realizing, they would ask me this question: sir, if sodium is continuously active everywhere in the body, then after a while all the sodium will go out of the cell and all the potassium will come into the cell? No. Sodium potassium pump is doing this all the time, but it is for counterbalancing some other movements, some other movements of sodium and potassium. They are being counterbalanced by sodium potassium pump. Remember this always. You cannot see it in isolation. Okay, so here is one more counterbalancing effect. Recharging of the excitable cells. Look, excitable cells, let's say nerve, its membrane excites. Means what? Depolarization. Sodium comes in and repolarization. Potassium goes out. We will talk about it later in the next chapter. But this is what the membrane excitation is. Now, this has happened in the excitable cell. The sodium potassium concentrations are disturbed. They have to be re-established. And therefore, after every impulse, sodium potassium pump will do what? It will throw the sodium out and it will bring the potassium back. And that's called as recharging. I mean, imagine our mobile charger. Suppose you have charged the phone and then continuously thereafter the phone keeps on coming. After a while, the battery will be drained out completely if the phone rings continuously after you have charged it. So, similarly, over here, if there are impulses after impulses after impulses, and as I said, after every impulse, sodium comes in and potassium goes out. So, after 100 impulses or 1000 impulses, what is likely to happen? All the sodium will have come in and all the potassium will have gone out for repolarization. And therefore, then the 1001st impulse, 1001st impulse will not be possible. So to avoid that, what happens is, after every impulse, whatever sodium has come in for depolarization will be sent out by the sodium potassium pump, and whatever potassium has gone out for repolarization is brought back in, and that process is called as recharging. Just compare it with the cell phone which I told you just now. After every impulse, sodium potassium pump has to recharge. And what is the meaning of recharging? Re-establishing the concentrations of sodium and potassium which have moved during the impulse. Okay, next, contributes in the process of BMR, basal metabolic rate. Look, what is basal metabolic rate? Also sometimes referred to as basal energy expenditure. Look, I am speaking right now, it is spending some ATPs. I am standing, anti-gravity muscles are spending some ATPs. I talk, walk, I mean each function needs ATPs. But even if we were not to do anything of this, not having taken meals for last 8 to 10 hours, lying comfortably in bed, no extra activity for any cell, still the ATPs are being broken. That is for the maintenance of cells. And much part of it is by the sodium potassium ATPase. It's a pump, it's an ATPase, it breaks down the ATP. And that breakdown of ATP at a very basal level is called as BMR. So, out of the total basal metabolic rate which is going on for the sake of maintenance of the cells and survival, 40% is by sodium potassium pump. Of the total basal metabolic rate or basal energy expenditure, 40% is spent on the sodium potassium pump alone. This is for all the cells. In the case of neurons, it is as high as 70%. 70% of the neuronal ATP breakdown under basal conditions is by sodium potassium pump. The next function is that this pump is electrogenic. That because of this pump activity, certain electrical charge gets created on the membrane, on the inside. How much? -4 millivolts on the membrane. If I say resting membrane potential of a muscle is -90 millivolts, it means inside of the membrane there is a charge of -90 millivolts. Of this -90 millivolts, -4 is contributed directly by the sodium potassium pump. -86 is by something else, which we will discuss in the next chapter, which is RMP, action potential. But why minus? First of all, why electrogenic pump, sodium potassium pump contributes minus charge? That's very obvious. Imagine a state of electroneutrality. Okay, this is just a hypothetical situation that I am asking you to imagine. State of electroneutrality, number of positive and negative charges is equal, let's say. That's the starting point. And then sodium potassium pump. Look, it is doing what? It throws 3 positive charges out, but only 2 positive charges are coming back in. 3 sodium out, 2 potassium in. Which means what? Which means slowly inside the cell, 1 positive charge will become less and less and less. And that means relatively negative charges will become excess as compared to the positive charges, right? So how much will that be in terms of quantification? It will be -4 millivolts, which is contributed by sodium potassium pump on the cell membranes, all cell membranes generally, right? So these were the functions. Now finally we will see the stimulators and inhibitors of the sodium potassium pump. Well, let's start. Aldosterone is a stimulator of the sodium potassium pump. It increases the number of sodium potassium pump. I need to explain this. Look, what is the function of aldosterone? It increases the sodium reabsorption. Let's say here is a renal tubular lining cell, and here is a sodium in the renal tubular fluids. Renal tubules, the lumen has sodium. Now sodium will be reabsorbed, means it will go into the blood vessel. This is the blood vessel. It is going to cross 2 membranes. On the apical membrane, there is a channel, for instance, ENaC, or there can be other transport proteins like NKCC or NCC or ENaC, epithelial sodium channel. So sodium goes in to the cell, and on the basolateral membrane there is sodium potassium pump. What is the job of sodium potassium pump? Remove the sodium from inside the cell and send it out. So this is the sodium potassium pump over here. It sends the sodium out. Therefore, the sodium concentration inside the cell will decrease, and because of that, the sodium will diffuse into the cell. Remember, I discussed some time back, the epithelial cells are polar cells. Why? Because of this very nature of the activity in their membranes. So sodium thrown out means cations become less inside the cell, anions become more, and therefore sodium will diffuse by the electrical gradient or electrochemical gradient. So that's how this works. Where is the role of aldosterone in this? Aldosterone keeps the ENaCs in the membrane for longer durations by inhibiting the ubiquitins. We have seen this. And on the other side of the cell, there is sodium potassium pump. So aldosterone will act here, and it will increase those numbers of sodium potassium pump. And this is how aldosterone increases the sodium reabsorption. Thyroid hormone increases the activity of the pump. thyroid is a major controller of the BMR. How does it achieve that? One way of controlling the BMR is by increasing the activity of sodium potassium pump. Then catecholamines stimulate the pump, especially in the muscle. Insulin also is known to be the stimulator of the sodium potassium pump. So these are the stimulators of the pump. Inhibitors, all these, remember, all these are the inhibitors of the sodium potassium pump. For instance, digitalis, we saw this some time back. Digitalis binds to the pump and inhibits the pump. Some diuretics are also known to inhibit the sodium potassium pump. That's how they cause natriuresis and diuresis. So some diuretics are known to do that. That was another D. Then dopamine. Dopamine inhibits the pump in the kidney. And therefore, this is the basis for giving dopamine in the conditions like heart failure. Look, on one hand, dopamine will stimulate the heart, pumping activity of the heart. The other action will be, it will inhibit the sodium potassium pump in the kidney. So see this renal tubule. This is a sodium potassium pump. If it is inhibited, what will be the result? Sodium will not be reabsorbed. That means what? Sodium will be lost into the urine, and that means what? Water by osmosis will be lost into the urine. So by doing this, dopamine will also decrease the ECF volume. It will cause natriuresis and diuresis by inhibiting the sodium potassium pump in the kidney. Another chemical which is called as the poison for sodium potassium pump is DNP, dinitrophenol. Dinitrophenol is also a poison or inhibitor for the sodium potassium pump. So this was a summary for the sodium potassium pump, but let me tell you, when we discuss various systems, you will get to know how important this pump is. I mean, in the CNS, we will describe penumbra, a particular ischemia, and the subsequent defect is called as penumbra. What is the root cause? Failure of the sodium potassium pump. Lots and lots of places. Overdrive suppression in the heart. What's the mechanism? At the root of the mechanism will be sodium potassium pump. So this should be understood in great detail, and therefore I thought I'll discuss it in the end. So that's the completion of the first chapter.